All right, I lied. We're not doing compaction. I thought the next one was compaction. It's implantation. I think the compaction stuff we talk about when we start talking about kind of juxtacontractors and paracontractors and the things that hold cells together or signal between cells, etc. So implantation. So we talked about cleavage up through the obviously this is only for mammalian systems or for mammalian reproduction because amniotic eggs and things, there's no implanting into the uterine wall or anything like that because there's no uterus, or they're not in there at least. So this is again our chat GPT image. This one looks kind of okay in terms of somewhat resembling something being implanted in a, looks like a cell wall. So yeah, good. All right, moving on. So this is an image of the blastocyst digesting a small wall or hole in the ZP, the zona pellucida, and then squeezing out or hatching almost from it. So that's kind of we talked about it in the last one. This is kind of how it's progressing here. So this is, you can see the cells here entering kind of the uterus. This isn't a mouse, so it looks kind of small. So this is the escaping that ZP is done with this digestion with a trypsin-like protease, which is secreted from the trophoblast, which digests that ZP and allows the egg to quote unquote, or the blastocyst to quote unquote hatch out because it's not needed anymore. Because remember, that's just to prevent, primarily to prevent implantation in the fallopian tubes and the ampulla before it gets to the uterus. So hatching from the zona and implantation of the mammalian blastocyst. This is, you can see the trophoblast here, which again, this becomes the placenta, and essentially it's the placenta. And then this is the intercellular mask, which will become the embryo. This is the initial implementation of a rhesus monkey. And this is the blastocyst here. So the endometrium, so the lining of the uterus, right here. So this says endometrium for those that are blocked by my camera here. So this has been modified by an estrogen and progesterone at this point that creates this extensive extracellular matrix. And that's what's responsible for this extracellular matrix is kind of a tacky or substance or I guess tacky is a good word. So it catches the trophoblast or the blastocyst as it comes into contact. So it's kind of like sticky, right? So it sticks it to the uterine wall and allows this implantation to occur. Let me get to my next slide here. So this is a little bit kind of a better diagram. So this is the tissue and germ layer formation early in the human embryo. So you see we have our blastocyst right here. The blastocoel is kind of the inside here. We have the trophoblast around here. The hypoblast is the inner cell line, and the epiblast is kind of the outside there. So at days 5 through 9, which would be up at the top here, is where implantation kind of starts. The inner cell mass delaminates hypoblast cells that line the blastocoel. So we start to get this kind of delineation occurring. And this forms the extraembryonic endoderm in the primitive yolk sac. So the epiblast and the hypoblast here. And you can see the implantation where we're starting to kind of penetrate here into the uterine wall. So at days 10 through, sorry, let's continue to point out some things here. Um, so this forms the extra embryonic endoderm, and you can also see here that we get this syncytiotrophoblast being formed here, like in spreading out into the uterine wall, and this growth of the cytotrophoblast, kind of a more closely proximity. Wow, I cannot get that word out. In close proximity to the blastocoel here. And you can see the syncytial trophoblast still exists here. And we're getting this amniotic cavity, so the amniotic fluid will start to develop right around here, where this amniotic cavity will kind of grow from this implantation site there. And then we move down to days 10 through 12. So the trophoblast divides into the cytotrophoblast. So we already got kind of this in this area. The numbers aren't lining up exactly right, but you get the idea. So it divides into the cytoblast, which will form the villi, and the syncytiotrophoblast, which will ingress into the uterine tissue, and this forms that chiron. So the syncytiotrophoblast are what helps to kind of bring everything in. So you can see it started forming up here, and this brings everything along. It kind of pulls it into the uterine wall. So in days 12 through 15, that is when we start this actual gastrulation, which we're going to talk about in the next unit, in the formation of the primitive streak. Then we'll proceed on to the notochord formation, the blastopore, and the additional organogenesis and things of that nature. Let's see, what else have I missed? The amniotic epiblast, right here, that's where the cells of the ectoderm, endoderm, and mesoderm, and the germ cells, which are kind of part of those, is like a small group of those three, smaller group. The main layers of tissue: ectoderm, endoderm, mesoderm. The germ cells are kind of a small subset that help for reproductive purposes in the future, so they're not super developmentally on the level of those three. They're kind of a separate little class. So we really think there's three, and then 4 little lowercase 4 types of cells. Anyways, those all form from the epiblast, embryonic epiblast. Oops, let's get my thing here. Right here. So the extraembryonic endoderm, where are we on here? Extraembryonic endoderm here forms that primitive yolk sac. So the actual size of this embryo at this stage would be the size of like a period, like at the end of a sentence, like a punctuation mark. So that's how tiny we are talking at this point. So it's like, even though it seems like we're growing big compared to, relative to some of our figures, it's still tiny at this point. A lot of information on this slide, a lot of things to kind of go through, but I think I covered the key points for you. Okay, so I edited this at some point. So this is the amnion structure and cell movements during gastrulation. So we'll get more into gastrulation going forward. So when, let's see. Do we want to cover this now? This is kind of a product of me changing the order of these lectures. I think we're going to skip this for right now, and we'll come back to it when we talk about gastrulation, because some of these things, like the primitive groove and things of that nature, we are going to you haven't been exposed to yet. So I guess what I will point out at this point, there will during implantation, we are going through gastrulation, and we have this amniotic cavity in this yolk sac here, the epiblast and hypoblast that we had talked about previously. And we'll start to see migration of cells. So we'll get this primitive streak development, and cells will migrate from the interior or from the exterior to the interior, start forming the tissue layers that are required for organ development. And so the first segregation of cells within the inner cell mass form two layers, which is the primitive endoderm, which I have labeled here because it's mislabeled here, the primitive endoderm and the epiblast. And so this forms what's called the bilateral germ disc. Like I said, we'll get a little more into this when we get into the gastrulation part. But this happens, for context, this just happens at the very early stage of implantation. And we're actually going to pause on this stuff as well. So we're going to actually cut this lecture at this point, and we'll cover this. I'm going to move this to the gastrulation lecture. So with that, that's a quick primer on implantation. We'll get more into some of these mechanisms. Just know that they happen kind of really early in the implantation stage, and we'll talk about them more with the germination or the gastrulation lectures. So I'll catch you in the next one.